Marlen, and I'm excited to talk about a project that I've worked on and put together with my mentor over the summer, and we're currently collecting data for the lasting effects of meditation on academic-related cognitive outcomes. Students experience unique and significantly high stressors in university settings. This can be because of multiple responsibilities that they have between school, work, and at home, and responsibilities that can be the lack of resources at the university. But these higher levels of stress can impact their physical, cognitive, mental health, and academic outcomes. The stress causes them to perform worse cognitively, and it can impact their grades or performance in classes. This can also be compounded in diverse and disadvantaged students population who already experience additional unique stressors. Meditation is an effective way to improve students' academic performance and reduce stress by using focused attention meditation, that's also known as FAM. What FAM does is it instructs individuals to focus their attention on a particular object. Normally it's their breath, and return their focus to that object when their mind wanders. This can improve cognition after a brief session in individuals who have never meditated before, making it a good intervention for students before they attempted to reduce their stress levels and improve their cognition function to do well in academic tests or responsibilities. However, it's unclear how these effects last, so it could be up to 60 minutes, but studies out there due to testing effects, because they use the same measurements to test the students' abilities at 20 to 40 minutes meditation. It may be because they're still learning the test. Our goal is to see if FAM affects memory, attention, verbal fluency in students both immediately and after 60 minutes completing it, while controlling confounding testing effects. It also benefits because we're looking at the multiple cognitive functions in memory, attention, and verbal fluency, while other studies have looked at one cognitive function. Our hypothesis is that FAM meditation session will enhance attention, memory, and verbal fluency in students immediately after and 60 minutes after its completion. To do this, we're currently recruiting 70 participants. They must have no psychiatric disorder, neurological disorder, eating disorder, head injuries, learning disabilities, and be pregnant or lactating. Our hope is that we may be able to extend to these populations after we get our baseline results. They are required to be university students, even community college students, between the ages of 18 and 55. And to control other variables that may affect their blood pressure, we measure and cognitive functioning. We ask them that they come when they get at least 7 hours of sleep the night before, and refrain from any caffeine, alcohol, smoking, and exercising 30 minutes before. We have them do a couple things. One is they do FAM or sham meditation. That is to count their breath. If they lose track or their mind wanders, they will go back to counting their breaths. This comparison is a sham meditation. Half of the participants are assigned to FAM and sham. A sham will tell them to sit and breathe if their mind wanders is fine, since it's a common comparison for FAM meditation. They will complete a visual analog scale, that's also known as VAS. That's a line where they indicate their current level of stress and blood pressure to get their stress levels. And they will also do their RBANS. That's a repeated battery neuropsychological status. That is multiple versions, A, B, and C, that test their memory, verbal fluency, and attention, that controls for their testing effects for different versions. So what they do is that they will come in, they will complete their baseline stress VAS and blood pressure to get their baseline levels and their first RBANS. Then they will do a 10-minute of randomized assigned FAM or sham meditation. Immediately after, they will complete their VAS, blood pressure, and complete RBANS once again. It should only take 15 minutes. They will watch a 45-minute plant documentary. That's it. We do the VAS, blood pressure, and RBANS one more time to get their baseline stress, cognitive function immediately after the meditation has been completed. Our plan is to analyze this data using 2 groups by 3 times. That's before, immediately after, 60 minutes after, repeated measures on ANOVAs on RBAN's memory scores, attention scores, verbal fluency scores, VAS stress scores, and blood pressure to see if meditation is affecting stress and cognitive functions. In conclusion, we are still looking at data now. I don't have any results to show you, but we hope by doing this study we will be able to clarify what cognitive function FAM specifically targets and how those effects last in the students. And what's great is that collecting data is in underrepresented students lays a foundation to test if marginalized students may benefit more as they experience more stressors. And I just want to give a quick thank you to my mentor, Dr. Carvain, and our lab research assistants, and the participants who are able to make this research possible. Thank you.
Thank you, Marlen, for your presentation. Our next speaker is Michelle Saint, also from Cal State Dominguez Hills, from the College of Natural and Behavioral Sciences, this time from my old department, although at Cal State LA, chemistry. Her title is Investigating the Impact of Electrical and Heat Stress on Human Cell Lines, a Biomarker and Biophysical Approach. Michelle.
Hello, I'm Michelle, and this is my presentation investigating the impact of electrical and heat stress on human cells. This experiment aims to investigate cellular responses to applied electrical heat and electrical and heat stress. After applying stressors on the cell's biomarkers, measurable substances in the cells, like heat shock proteins, and biophysical properties, physical traits, like the cell membrane integrity and morphology. They are, we measure them to see how cells can adapt to such stressors. And we hope that this experiment will clarify how cells adapt to these stressors. The importance, relevance, we have chosen to study this because we are interested in expanding our knowledge on cellular biology. But this is also practical for the medical and pharmaceutical industries, where we can hopefully our research can contribute to therapeutic strategies that might help patients without stress-induced damage, like chemo, for example. There is potential to improve existing treatments. Results can also provide insight for the biotechnology and agricultural fields, like creating stress-resistant crops and making food sources stronger and more resilient. And finally, public safety is always a major concern. Understanding how cells respond to electrical and heat can help establish evidence-based guidelines to protect us humans from extreme temperatures. And our objectives are to analyze heat and electrical stress on various human cell lines. We have 8 different cell lines that we're studying, observe how these cells respond to stressors, and identify potential pathways to prevent these responses. Methodology. So we are conducting this experiment by applying stressors on these 8 different cell lines.
Michelle, I don't see your slides moving.
Oh, they're not?
Now they are.
Okay.
Thank you.
The stressors are applied. So we have electrical and heat stressors. We're testing electrical because it's often overlooked in these sorts of experiments. And electrical stressors, they are applied via a electroporator apparatus, like the one on the left. And essentially what this does, it penetrates, it sends pulses of electricity that will penetrate the cell membrane. And before we conduct any real experiments, we are going to be conducting preliminaries to optimize these values, because we need to optimize the voltage and pulse duration ranging from 100 to 600 volts to and 10 to 100 milliseconds in order to ensure that we aren't killing off many of the cells and we have adequate stress. What is it called? Responses. Heat stressors are applied to cells through a heating block. And we are going to be exposing them to elevated temperatures outside of 37 degrees Celsius, which is where we maintain them in our incubator. We are going to be measuring biomarkers, such as protein quantification. Through protein quantification.
Michelle, you're still on the second slide. It says methodology. Are you changing your slides? Do you want them?
Yes, sir.
Okay, thank you.
So biomarker measurements. We are quantifying proteins. We're specifically looking at heat shock proteins, which are responsible for refolding proteins when they're damaged by heat. These are quantified using ELISA kits, enzyme-linked in immunosorbent assays, and we're also going to be using Western blots. The ELISA kits, they provide quick results, but Western blots, they will quantify the results from the ELISA kits. Also, gene expression analysis, we will be quantifying mRNA levels, specifically of genes related to stress-related biomarkers, using quantitative polymerase chain reactions. And RNA will be extracted from the cell cultures and quantified using SYBR green. And then we will also be measuring reactive oxygen species and nitric oxide levels within the cell. To analyze the biophysical properties, we will be using a live dead viability cytotoxicity kit, and this will help with measuring the cell membrane integrity under stress conditions. Chalcedone AM is used on live cells, and propydium iodide is used on dead cells. Phase contrast microscopy will be used to analyze changes in morphology. Every single day we take pictures, we record the shape, size, adherence of the cells when we've been cultivating. We'll also monitor the calcium levels, as these indicate signaling in response to stress. And we'll be using fluorescent indicators on live cells. We also have a dye called JC1, which will be used to assess the changes to the mitochondrial membrane potential. And this dye will change colors as the mitochondria becomes more or less polarized, which can help us distinguish between healthy and functional mitochondria from depolarized ones. The collected data will be analyzed using statistical software, and p-values less than 0.05 are statistically significant, which will represent a meaningful difference in expression induced by these stressors. In the near future, our experiment may be hindered by contamination. Just to clarify, we haven't started. We've been cultivating these cells. It takes a while. They may be hindered by contamination to our growth medium, because in order to grow them, we need to have a medium for them to eat. And these can cause cells to die to infection. Equipment may also malfunction, or it may run out of material for our experiments. Thus far, we have seen some of these challenges. On August 14th, our CO2 incubator was forced to halt experimentation until it was replaced. We address these challenges in various ways. The lab maintains strict safety protocol to keep our cells safe. We also aliquot master mixes in case of potential contamination to our mediums. And we've been cultivating numerous cell lines for a few months, mainly UMR 106, to stock up for our experiment. And I would like to acknowledge Dr. Horace Krogman, the primary investigator and my professor, and Gisela Alvarez, his main research assistant and my colleague. Thank you.
Thank you, Michelle. Our next speaker is Axel Tristan from Cal Poly Humboldt, the Department of Wildlife, Amphibian Pathogen Surveillance, and Basal Detection in the Arcata Community Forest. Axel.
Okay, can everyone see that okay?
Yes.
So hello, good morning. My presentation is going to be on fungal pathogen surveillance in the North Coast salamanders. So some quick facts. Chytridiomycota or chytrid fungus are zoosporic fungi with motile flagellated spores which move independently in water and on land. Batrachochytrium dendrobatidis, or BD, affects frogs and toads, causing roughly 90 extinctions and 490 population declines globally. Batrachochytrium salamandrivorans, or BSAL, is an evolutionary offshoot of BD, affecting salamanders and newts, and has been found in Europe and Asia, specifically the Netherlands and Southeast Asia. Our goal with this is early detection and establishing accurate population dynamics for the West Coast. So here's a map of our study area. We utilized a systematic sampling design with the help of historical data collected from previously participating students at Cal Poly Humboldt and generated roughly 50 sites in our study area to conduct our swabbing procedures. Our first point was randomly generated, and each of our sites are roughly 400 meters apart. Professors David Sin and Richard Brown are working with us in establishing accurate population amphibian surveillance protocols and methodologies because we need to better understand amphibian populations in our study area before BSAL arrives in this region. So this is the same map from the previous slide, but each of these sites has been singled out as they all had historically recorded a particularly high abundance of salamanders in each plot from previously involved student researchers. Okay, so why is BSAL so scary? Well, similarly to BD, a majority of amphibians breathe through their skin, roughly 2/3 of salamanders and newts, which means that chytrid fungus essentially suffocates them to death, similar with BD in frogs and toads. So BSAL impacts the keratinized layer of skin and may also have neurological effects on salamanders and newts. Some common signs and symptoms of a chytrid infection in a salamander or newt are skin lesions, abnormal behavioral changes, lethargy, and inability to move. So as part of our study, we will be collecting biological samples via bodily swabs on the ventral and dorsal sides of the salamanders and newts, and as well as collecting any post-mortem specimens that we determine may have been deceased by a BSAL infection. So these are some slides, some pictures on this slide and the last slide of what BSAL infection can look like in salamanders. There are black border lesions as well as lethargy and due to inability to breathe. That's on the left side. On the right side, we have a graphic taken from the SNAPS handbook, which is the Student Network for Amphibian Pathogen Surveillance. We are working with them to collect this data and report this. So this graphic is demonstrating the regions on the salamander's body where we are supposed to swab 5 times on each portion in order to collect the best possible chance of detecting BSAL infections in an individual. So one of the many measurements and biotic features we are required to collect for SNAPs is the snout vent length, or SVL, which also offers us a great opportunity to monitor the individual and observe the individual for lesions and other signs of a potential B-cell infection. So we originally started with 2 specific focus species for this project based on susceptibility to B-cell and abundance, population abundance in our study area. Those being the one pictured here, which is an Oregon ensatina, and rough-skinned newts, which will be shown on the next slide. This Oregon ensatina here pictured did not display any of the established symptoms of chytrid infection. It was very mobile, alert, and did not have any visible skin lesions or lethargy. So like I mentioned earlier, we are working with our advisors, Richard Brown and David Sin, as well as the SNAPS organization, Student Network for Amphibian Pathogen Surveillance. We established sampling sites in the Arcata Community Forest, and we are swabbing local populations in the ACF, which is roughly 40 individuals. In the future, we plan to expand our sampling sites beyond the Arcata Community Forest and expand our number of study species, as well as gathering a larger sample size. So these are some of the focus species that I mentioned earlier. These are 3 adult rough-skinned newts that were recorded for our study. Along with these 2 focus species, those being the ensatinas and the rough-skinned newts, SNAPS also has a myriad of other local species that are here on the list for data collection, should we run across them, and that's what you'll be seeing in the next few slides. And then these are some 3 different juvenile coastal giant salamanders that we swabbed in a creek near campus in the Arcata Community Forest. These guys are pretty popular in the world of herps due to their size when mature, as well as their coloration and unique patterns. So these are a great example of 2 sub-adult northwestern salamanders that we swabbed in the Arcata Community Forest. Both were very healthy and responsive and much easier to handle compared to some of the other species we had encountered. And then last but not least, we have the small but mighty slender salamander. These are 2 juvenile specimens that were swabbed and released in the Arcata Community Forest. Although this is not a comprehensive list of all of our local swabbable salamander species, this representative list covers quite a few of the more commonly encountered natives. I would like to acknowledge and extend my deepest gratitude to my girlfriend, Lindsay Mounts, for being such an amazing volunteer and co-contributor on this project to help me collect data and swab all these amazing individuals. I would also like to thank CSU STEMnet for the primary funding for this project and the opportunity to present here today, as well as the HLA Ghana Scholarship Foundation for secondary funding. Finally, I would like to extend my utmost gratitude to my advisors, Professors David Sin and Richard Brown, and of course, the SNAPS organization for allowing me to contribute to their nationwide project. Thank you.
Thank you, Axel. Our next speaker is Ashley Robinson. She's a junior from Cal State Fullerton, Department of Physics, and her title of her presentation is Harnessing the Collective Motion of the Nematode T. aceti to Produce Work. Ashley.
Hi, good morning. I hope everybody can hear me well. So actually, hold on, let me go back, sorry. So yeah, I am working with the nematode, also known as the vinegar eel. And so for a little bit of background on nematodes, sorry. They actually, based off of previous work done by my research professor and my advisor, have been found to actually synchronize their oscillations or just participate in collective motion. But the really interesting thing about them is that this is only when they're placed into certain areas of confinement where there's a contact angle that's less than 68 degrees. So you can kind of see it visually in this bottom image. We have 80 degrees, 60 degrees, 45, and 30. And these are just little triangular pockets that have these varying angles, and this is their synchronization. You can see that in 45, there's a little bit of synchronization, but in 30, it's almost perfect. They kind of look like one nematode. And then here's just some images to show like visually a better representation. So there's a single little nematode in the corner over here. And then we have a video showing their synchronization. And so my goal is to be able to replicate their conditions of confinement. So when they best produce the synchronization. And by doing that, we are taking 3D designs to replicate a water droplet, but in a certain way in which we can kind of control it. So right here, we have this microchannel down here, and there are some triangular pockets that you can see that kind of go off on the side. And so that's where I'll put those varying angles, degree angles, and they'll be synchronizing. So if they synchronize in these corners and they start to collectively move, we can assume that they're exerting some form of a force in the middle of the track. And so the goal here is that using that force, we can then kind of create an on-demand fluid flow, right? So they'll start moving either clockwise or counterclockwise. And then we are also using microboats. So microboats are just going to be little triangular wedges that we also put certain angles in so that we can kind of hope to get nematodes to synchronize in between that angle and then push the object around to track object displacement, which is just another way of measuring the fluid flow. So here we go. We got a little process, a little video. I hope you can all see this pretty well. So this is just to demonstrate what my experiments really are. So we just start off with a collection of a sample, and then we put that in a centrifuge, get a really dense, more dense sample, and then we place that within the 3D object or on a water droplet, and then we place either the microboat or, like I said, just in a 3D object. And then what I do for the microchannel to measure fluid flow is I place tracer particles within it. So that's those little dots that you're seeing in this experiment. So if I can get those moving in a certain way, then I can easily track their velocity or also object displacement throughout a couple of frames. And so we got a couple of results here. Unfortunately, after we had started with the microboats, we actually ran into some issues. So we 3D print our objects using resin, and we actually found that it was really hard to get the correct density. So if I were to put the droplet on a slide and then place the microboat inside, it would either flow to the top or sink to the bottom. There was no real way for us to really get that perfect density to keep it in the middle so that the nematodes could actually group behind it. And we also tried to mitigate this by placing it on not a hydrophobic slide, just a regular slide. And then we would control the droplet within a well, which is this little circular object that you see right here. But that also caused leakage. So we kind of had to say goodbye to the microboats for a second. And we moved on to the microchannels. So you can see an experiment up top of the microchannels. And then on the bottom, we're actually tracking the object's velocity. So the bottom left-hand picture is going to be the first 5 frames of this experiment. And you can kind of see these little lines right here. These are going to be where the objects were going within the first 5 frames. You can kind of see that it's sporadic. It has no real direction. And then in the last 5 frames, we got almost perfect direction, pretty synchronized. And so we got a recorded average velocity of 0.026 millimeters per second on that one. And then we actually noticed within this design, the flow was generated, but it was actually going in the opposite direction of what we were hoping to find based off of where we were putting our triangular pockets right here. So if I wanted it to go clockwise, it would go counter and vice versa. So we decided to take on a new design, which is by curving the walls of the microchannel itself rather than just cutting straight through an object. This is because it lessens the potential for unwanted synchronization, right? So if I were to cut into that object, it would actually create more angles for the nematodes to synchronize in, which would cause, we were hoping to see that it would cause this counterclockwise or clockwise flow, the opposite of what we wanted. So then, yeah, we curved the walls, hoping that this would mitigate it. And then we actually ended up finding really cool instances throughout using this design. So the first one in the top right-hand corner is we actually noticed there's microsphere alignment. So rather than moving across the channel, they were actually frigid and stayed very stuck in the middle of the channel and in somewhat of a linear formation. And then if you look at this left-hand video, play it again. I mean, sorry, right hand video. There's microsphere formation going on right here, which is really interesting. I think I saw the number 4 in there at some point, but we thought that was a really interesting instance to explore. And we also have down here on the bottom a rotational flow being recorded. And what I'm using for this image to the left of it is image day. And I'm just tracking the particle displacement, sorry. And we can see over a couple of frames, there's this rotational flow going on. And so you might be wondering what the applications are for, trying to see what these fluid flows can do, or these nematodes can do for their fluid flows. This is basically to gain control over collective entity systems. So just being able to generate on-demand fluid flows tells us a lot about fluid flows and the naturally occurring phenomena in general. But this can also be applied to engineering of synthetic active matter systems with broader applications. So things like targeted medicine delivery and just being able to replicate active matter systems through engineering. And I would like to thank STEMnet CSU for the funding for this research, this summer research. And I would also like to thank the people in my group. So I have Christina, Evan, and then my research professor advisor, Anton Peshkov. And lastly, I would like to thank this little guy down here who made it really hard to keep control of my density because he kept eating them. So thank you.
Thank you, Ashley. Our next speaker is Javier Villalbazo from Cal Poly Humboldt, the Department of Biology, Department of Zoology, excuse me, from the College of Biological Sciences. Tolerance to copper-based toxins in invasive bryozoans in the genus Recipients. Javier.
Howdy, everybody. My name's Javier Villalbazo. I'm a Humboldt native. I'm from Hooper, California, and I'm a zoology student up at Cal Poly Humboldt. Over the summer, I had the pleasure of working with grad student Dana Johnston and fellow undergrad student Abigail Price with the oversight of Dr. Sean Craig of the zoology department. We were researching the costs and trade-offs associated with tolerance to copper that the invasive species water cipra have evolved to have and allowed them to become invasive and attached to the bottom of holes. And Dr. Craig is an expert on bryozoans, so it was really awesome to get to work with him. So what are bryozoans? Bryozoans are these really awesome invertebrates that are colonial, and they're found in freshwater and marine environments. They're lophophorates, so they have this lophophore feeding organ that they extend out into the water column, and they collect their food particles and bring it back into their body. And they don't require much energy, so they're able to maintain this sessile modality of life, and they make it work for themselves. However, they can be outcompeted by ascidians and tube worms for space. That's just to name a couple of things. So those are some issues that we ran into when conducting this experiment. The bryozoans that we are doing our research on happen to be from the marine environment, and they resemble a red potato chip. And when clustered up, they become this like beautiful arrangement. So there's 3 things to introduce you to. These bryozoans are, they've evolved to be resistant to copper-based toxins, which are very commonly found in anti-fouling paints. Anti-fouling paints are things that stop things or stop organisms from settling on the hulls of ships. So this resistance, though, is thought to have a cost, and that is the basis for what we're researching. As a result of the copper tolerance, bryozoans have been known to be an invasive species, and they have an impact on the ecology of wherever they go, but again, they can be outcompeted pretty easily by other things. However, in scarce food, in an environment that has a scarcity of food, it can be a pretty major impact. So just to reiterate for copper tolerance, vessels, both small and large, use anti-fouling paint, which is composed of compounds meant to hinder the attachment of organisms to vessels or outright kill larvae and adult organisms that attempt to settle on the vessel. And this is a pretty successful strategy for barnacles and other sessile invertebrates that exist in the ocean, and their larvae swim in the plankton and settle on things naturally. A major component of the anti-fouling paint is copper. Said that. Yeah, so water cypra have evolved to be resistant to this, and as a result of that, they travel on these vessels wherever they go. No matter if there's a vessel here in Humboldt that's traveling to Portugal, they are highly adapted to needing little food and being able to survive on these highly toxic hulls. So invasive species, again, reduce biodiversity by outcompeting native species with limited food sources. What also happens in the vessels that bryozoans are attached to, along with other invertebrates, are that they make the ship become less efficient at moving. And this increases fuel consumption and time to destination, which is a pretty big deal for shippers and freight movers, and it accrues a pretty big cost. This slide here has some of like the Hall of Famers in terms of, the Hall of Famers that collect on the bottom of things or invasive species, such as the zebra mussel, which outcompetes quite literally everything, and barnacles, Darwin famously hated. So our hypothesis for this experiment is that adult water cypra exposed to copper will show lower growth rates and fecundity, meaning they'll produce less offspring, with morphological changes that result in smaller and fewer zooids and narrower apertures. Apertures on this diagram we have here of Ascophora, which is another grouping that water cypra are in. The aperture is that pore that allows water to come in, and it inflates that sac when the parietal muscles contract, and that allows for the lophophore feeding organ to come out. And when water is expelled from that cavity, the lophophore comes right back inside the organism. The smaller aperture would be beneficial in reducing the organism's contact with water that may contain anti-fouling paint. This paint is designed to come off at a very slow rate, but introduce it to organisms that are on and around the hull in order to prevent them from settling. Copper is very toxic. So in the marine lab at Cal Poly Humboldt, there were colonies that were gathered by a previous researcher, and they have known genomes and relationships to one another, so we were able to utilize those. We utilized the lab-dwelling bryozoans in order to clarify whether traits such as colony size, number of zooids, size of zooids, and reproductive outputs are traits controlled by genetics or are affected by copper exposure. The field experiment was done using panels that had dissected colonies placed near the center, and I have an example of the colonies, or sorry, the panels that the colonies were set on in the bottom there, which is a PVC panel with holes drilled into it that we were able to attach a bolt to, and we were able to screw a wing nut in there so that we could fit that to a frame. The panels either had no anti-fouling paint, mild copper content anti-fouling paint, and high copper content anti-fouling paint, which were, the mild was about 20% concentration and the high copper content was about 33%, but these anti-fouling paints can exist as high as about 75%. So, but those were extremely expensive. So we were limited in that capacity. The frames were suspended in the bay using PVC pipes that we had drilled holes into, and we were able to fit that bolt and wing nut in there to suspend them. The panels were reattached to the PVC pipe after every cleaning, and we cleaned every single week, and we were able to gather data every single week on these guys, measuring their growth and number of zooids. Fecundity is a little bit hard just because they release at variable times, but at a later date, we're going to be measuring fecundity using the knowledge that bryozoans release larvae when exposed to heat and in direct contact with sunlight. So this is an example of what the panels look like, and I have measurements from week 1 and week 2 for both of these, for number 33 and number 36 here. These two are siblings, and so they're pretty closely related. And as you can see, number 33 performed very well growing into the control panel in almost a random fashion and extending its zooids and its colonial area very far and wide on that panel in a short span of just 15 weeks. And as opposed to number 36, by week 15, it had reached about 2/3 the size of number 33, which is indicative of the colonies exposed to copper, though related, grew at a slower rate, and that would be because they accrued some cost in being able to survive coexisting with that copper paint next to it. So of the original 45 bryozoans that we dissected from the lab, 28 survived, and it was kind of expected that a few would die, just because it's a very traumatic experience being ripped from the colony where you're all collectively sharing a food source. And yeah, so there was thought to be some. And also in the first week when we placed the panels out there, there was a dredging of the water nearby, which caused basically suffocation of the colonies for an extent, and a lot survived, but it's understandable that a few died. So 11 were control panels, 8 were on the mild, and 9 were on the high exposure, which was interesting because you would expect the mild to have a higher survival rate than the high, but due to variable circumstances, this is the data that we got. We're yet to measure fecundity, and it was interesting that all survivors had at least one sibling or clone that also survived, and all of the zooids and colonies that died were of the same relationship or a clone. Yeah, preliminary data confirms the slowed growth portion of the hypothesis. However, there's still a lot of data analysis that needs to be done on this topic. I'd like to thank Dana Johnston and Dr. Sean Craig. Sean Craig isn't in these photos, but he was an amazing resource to get to learn more about these marine animals. And also Abigail Price, another undergraduate student that I got to work with, and she's also amazing. Also, thank you, STEMnet. The financial support helped quite a bit during this. And that's all I have for you. Thank you.
Thank you, Javier. Our next speaker is Taylor Galupi from Cal State Dominguez Hills, Department of Anthropology. And their title is The Evolution of the Invalid Alongside the Development of Paralysis Care, the Invalid, sorry, Unfolding Able-Bodied Mentalities on Disabled People. Taylor.
Hi, thank you so much for that introduction. Let's get right into it because, 8 minutes is pretty quick. I am mostly going to be comparing current paralysis care to the paralysis care of the 20th century. So as we know, as long as there have been people, there have also been impaired or differently abled people. But the idea of disability as we see it today is more so something that's a product of the Industrial Revolution. As factories became the main source of income for most people, and there became a standardized version of productivity, anybody that sort of lied outside of that productivity was considered a burden on this sort of new infrastructure. And it's that infrastructure that led to the mindset of people towards paralyzed people during the polio epidemic. And just as a baby background on polio, we mostly all know, but it is a virus that's spread through the fecal oral route, and it's not always paralytic, but it did paralyze around 35,000 Americans every year until around 1997 when the vaccine was developed. And the vaccine did wipe out pretty much all polio in the US, but there have still been cases of other nations that do have cases even today, such as in early September of this year, the World Health Organization actually started a vaccine program for children in Palestine because they don't have access to clean water right now. As for the method of my research, it's mostly archival with some interview. I've been through the CSUDH archives in the Harlan Hahn collection and through the USC archives. And the interview has mostly been with people working in care, whether it be patients or placement workers or just caregivers. And just to mention, this is sort of a background of what will eventually become a larger project on caretakers and their patients. So since I mostly use the Harlan Hahn project or Harlan Hahn collection at the archives, I want to talk about him as a person before I talk about him as a patient. He was a lawyer, a professor, a disability advocate, a father, and a disabled man. He actually was a USC political science professor who sued his place of employment while still working there for lack of accessibility. And it led to a huge change in infrastructure in the university that you still see today. And he was also a lawyer who was one of the people to push forward the US Rehabilitation Act in 1973, which was the first federal act forbidding any discrimination for people based on disability. It also improved accessibility in the medical center and made it to where it was required for a percentage of federal jobs to be given to disabled people. He was 6 years old when he was diagnosed with polio and was dealing with its effects up until his death at, I believe, 68. So as I had mentioned, Harlan was 6 when he received his prescription from the Mayo Clinic for one spoonful as needed of phenobarbital, which is a pretty intense sedative, especially knowing that the other main treatment that people were using for the paralysis associated with polio is soaking in federally sponsored hot springs. This was sort of seen as a half vacation home, half medical facility that was like a call to nature, sort of, maybe because of being so far from Earth and how we do things, going back to nature and being in these sort of God-given areas. This can be what heals these more modern diseases. And some of these waters were charged with radon to become radioactive to the point where they were banned from being shipped by the government, but the government still funded the actual hot springs that people would soak in these radioactive waters. There were also, of course, like very familiar treatments like soft tissue work and muscle manipulation that you still see for certain things today. And larger so, we can talk about the public attitude of people during this time period. It was really expected to sort of keep patients in the dark about their treatment, especially with instances of infantile paralysis, which basically just means anyone under the age of 18 who has some form of paralysis, and allied conditions, which can be anything from stroke to arthritis to anything along those lines. And it was also really encouraged to have as little interaction with the patient as possible, only touching and only moving when absolutely necessary, and almost sort of encouraging a forced solitude on patients, which didn't really have anything to do with contagion and just more so had to do with the social pariah. And on a more social note, the founder of one of these hot springs that were federally founded was quoted on informational booklets sort of advertising his baths, saying that he goes to his own baths, not because he is an invalid, but because he intends to never be one. And it's just sort of this idea of othering for people that have paralysis and people that have different bodies. And even FDR during his campaign, did everything he could to hide his diagnosis up until being elected, because he and his community were worried about his sort of disabled body equating a weak mind. So fast forwarding, we are talking about Rancho Los Amigos, which is one of the most popular and highly rated paralysis rehabilitation and research centers in the area. They're actually pretty close to my campus. They focus not just on the length of life, but the quality of life for people with paralysis and people with disability in general. And for example, I interviewed a man named Eddie, and we discussed his injury and how Rancho has sort of shaped his post-injury care. He was 50 years old when he was driving down the road and was victim of a violent crime, and that has left him in a wheelchair for the past 12 years. But he is also a member of Rancho's rehabilitation work program, and basically the way that works is they take sort of people that want to be more involved in the Rancho community, and they start them. For instance, he started in the cafe and has worked his way up to processing samples in the PK lab. And the importance of these programs to him is that he felt like he had received so much care, both on the emotional and physical level, that he really wanted to form sort of a deeper community with Rancho, and has even started working on setting up a chess campaign for helping people with limited mobility in their hands to sort of work on building their dexterity. So why bring any of this up? Clearly, in comparison with a few of the other presenters, I am a lot more qualitative in my research than quantitative, but that's kind of the point. Talking about qualitative research and sort of solidifying disability studies in anthropology and medicine is incredibly important and can sometimes be looked over. And it's more than just about diagnosing a symptom and figuring out a way to cure it. It's about viewing a patient as a holistic person that comes from whatever their background is and has to live every day, not just during your 15-minute doctor's appointment, and figuring out the best way to sort of have recovery, not just physically, but like with Eddie, mentally and community-based recovery. And on top of that, remembering that archives are huge for establishing precedents, just like Han's work, which was donated to CSUDH and was what led to him being able to get the Rehabilitation Act, which was what partially funds places like Rancho and Rancho itself. So I just want to say thank you so much to STEMnet for giving me the opportunity to work on this for the summer. And I also want to thank Professors Dr. Luxis and Dr. Armstrong and Archivist Tom Philo for helping me sort of dive into disability studies.
Thank you, Taylor. Our next speaker is Dino Santia, Cal Poly Humboldt, the Department of Forestry, Fire, and Rangeland Management. Title is Understanding the Impact of an Herbicide Treatment in the Sagebrush Steppe. Dino.
Thanks, Dr. Gomez, for that introduction. So, gonna get the slides to work. Okay, so the research project that I've been working on is focused on the recovery of a sagebrush steppe community that had been invaded by introduced annual grasses. This conversion of sagebrush ecosystems into annual grasslands is occurring as a widespread problem throughout the arid western United States. And it's problematic because annual grasslands have higher fire return intervals. They store less soil carbon due to a much less robust root system when compared to native perennial counterparts. There comes a loss of wildlife habitat, such as that for the sage grouse. And this conversion also leads to a decreased quantity and quality of available forage for cattle production, which is an important part of agricultural rural communities. To address this issue, land managers have several restoration techniques, a couple of which include the application of herbicides on the annual grasses and also the application of native plant seeds, both of which were applied to the study site that I will be looking at. And so data from this project can be used by land managers to help understand the impact of their actions and inform future decisions when trying to recover sites that have been converted into annual grasslands. Some specific research questions that I'm trying to answer with this study and with the data available to me are how the above ground annual grass populations have changed at my study site, or at the study site I've been working at, and also how the native perennial grass populations have changed at this study site. And I'm interested in these two broad categories of plants specifically because they're good indicators for overall site health and because the land managers have applied different treatments that were directed at influencing these two different groups. So the study site that I was working at this summer is located on a Bureau of Land Management grazing allotment just outside of Klamath Falls, Oregon. In 2013, this site burned severely, which was then followed by an invasion of an annual grass called Medusa Head. And then in 2020, an imazapic treatment was applied, which is outlined on this map in blue. Imazapic is a pre-emergent herbicide that specifically targets annual grass seed. And then in the fall of 2022, a native plant seeds were spread across the site to hopefully get reestablishment. The study that I've been involved with began in the summer of 2022 and is running along a 5-year timeline. I got involved with it in the winter of 2024. There are two major components to this study. One is the above ground vegetative portion, and one is looking at, and the other is looking at the seed bank at this site. And so during the summers, crews go out and visit 5 different sample plots, which are marked in purple dots here. And following the BLM's assessment, inventory, and monitoring protocol, vegetation data is collected using line point intercept methods. Along these transects, the soil samples are also collected, which are being used in the seed bank study. And also grass litter depth and soil moisture are collected along these sites. So moving forward, I'll be presenting some of the results from the above ground vegetative portion of this study. The seed bank study is still ongoing. Okay, and here is a figure showing the total vegetative cover across the 3 years that data has been collected, with time on the x-axis and percent cover on the y-axis. We can see that there's been a general trend of increasing vegetative cover across the 3 years of this study. And I think it's interesting to note that when looking at some broad groupings of plants, the majority of the increase has been in herbaceous, non-woody plant types, while the woody shrubs have contributed very little to the increase in plant cover. And now looking at some figures that are going to help address the research questions that I brought up. Again, we have time on the x-axis and percent cover on the y-axis. Looking at the annual, specifically the annual grass cover, we can see that increasing trend also happening. And this is the same with perennial grass cover. However, it's important to note that after running some statistical analysis, there was not shown to be a significant increase in annual grass cover due to the high level of variability in the data set that we collected, which is indicated by this large error bar here. And in contrast to this, there has been a statistically significant increase in perennial grass cover, specifically within this last year. So, from some of these results, it can be seen that the management practices are appearing to have a positive influence on perennial grass cover, which is a good indicator that this site is recovering from some of the disturbances and that the management practices are having a positive influence on the overall health of this sagebrush ecosystem. However, I think it's important that there will be continued monitoring of the invasive annual populations because of that increasing trend. And this can help the BLM office determine the need for any further management interventions. I also think that some more attention should be placed on shrub recruitment in order to continue the restoration of the site, because the data that we have collected picked up on very little recruitment. And so I'd like to end by thanking the CSU STEMnet program for sponsoring this research and giving me the opportunity to present to you all today. I'd also like to thank Dr. Carrie Byrne for supporting me throughout this process and giving me the opportunity to work on her research and mentoring me through the process. And I'd also like to thank Klamath Falls BLM for helping with the data collection. Thank you.
Thank you, Dino, and thank you to all of our great presenters today. It's always exciting to listen to students speak on their research, and hopefully this opportunity, this experience will lend you many more opportunities going forward. So we'd like to open the floor for questions. They could be certainly from the audience, and the audience is also the speakers. So I'm going to open the floor, and you can just raise your hand in the chat. I can't see all of you right now because of the way the Zoom's set up. But please, if you do have a question, and then state who it's for, and then you can ask it. Any questions?
Can I ask why the nematode to Ashley? Because I found that to be really awesome. That was a great choice.
I'm sorry, what was the question?
Well, why in particular did you choose to do the nematode? Did you have a professor that was already conducting that research, or do you have another path of thought?
Oh yeah, so the lab that I had ended up joining was already doing research or is centered around the nematode, but they're pretty fascinating enough to center your research around, I would say, simply because they can naturally synchronize their oscillations, so their body movements, they can synchronize them. And it's a naturally occurring phenomena, so we don't have to do it too much to be able to like induce this. And they're right now one of the only known species, at least of nematode, that can do that. So
That's amazing. Thank you.
Good afternoon. I have a question for Axel Tristan. What challenges did you face during your project?
Thank you for your question. I would say that one of the largest challenges that we faced when collecting data was definitely having to contend with weather. Amphibians are very moisture-based, and during the time that we were doing our sampling during summer, it was very hot and dry, and so available moisture in the soil was hard to come by, and so it was especially challenging to collect specimens to sample. I think we would probably do this study in the future in a more wet time of the year to be able to find more specimens more readily.
Thank you, Axel.
Other questions? Don't be shy. Okay, I'll ask one. So Ashley, okay, David.
Yes. Sorry, I'm getting my screen back up here. Mine's for Michelle. I was kind of curious about the application of heat shocking and electrical shocking on human cells. What's the application for that work?
So the application for that, well, first of all, academic interest. Electrical stressors aren't usually studied in stress tests. So that's an area that has a gap in knowledge, and we're looking to kind of fill that gap. There are also applications in the medical and pharmaceutical industries. Our research can be used in the near future to help create new therapeutic strategies that can potentially reduce the amount of stress damage that human cells take from modern day treatments right now, like chemotherapy. That's very intensive on the human body. I'm not saying that we can cure cancer, but potentially our research can help create an alternative towards chemo.
I could imagine this would help in or your research would help with infants who experience febrile seizure disorders, as well as like sepsis patients in the ICU. So those are very amazing applications.
So I have a question, and any of you can answer this. How has this experience in conducting research changed you as a person?
In terms of research, like it's very humbling, the amount of research that you can get done in a period of time. Like honestly, I think we all felt a little maybe Newtonian, like we could just go out there and change the world, but you have to set your parameters very narrow and focus on something in order to actually achieve real, definitive and meaningful results.
How about anyone else?
I also share Javier's sentiments. I definitely thought I had a lot more time, but the amount of research that we were able to get done during a few months is, although it's not that much, I'm looking forward to sticking on this project much longer. I really want to see the end of it. Also, this is a valuable experience for me, even though I don't really plan on doing much with biophysics in the future. My aspirations are actually in forensics, but I think this experience is still valuable because I've met very good colleagues. Like I acknowledged my professor, Dr. Horace Krogman, and his assistant, Gisela Alvarez, amazing people. It's great working with them. I also appreciate the lab experience that I'm getting hands-on because this is new technology that I wouldn't be handling in my current chem labs.
I'd also like to share maybe a little positive aspect, because humbling sounds a little harsh. I would say though that this really did help enhance, I would say, critical thinking skills. So being able to recognize, opportunity within discovery. So, in terms of my research, there were areas where we ran into issues, but then through working around that, we found a lot of other different things. And just being able to recognize, just all of the opportunities and all of the applications your research could have for other things. And even though I would also enjoy seeing the end of this, I feel like there's just so many opportunities that continuously come up where it's like, we saw something very interesting here, so we're going to continue to research it in this area and in this area. So I think it just really helped enhance critical thinking process and being able to really see where you can take things based off of what you're observing.
I'll also add on to that. I thought it was very interesting to see how kind of like how the sausage is made, right? From a perspective of actually doing the background work, and not just seeing or doing what you see like on TV, right? Where it's like a guy in shorts running around the woods and playing with animals. There's a lot of background work that goes into it, a lot of reading, a lot of uh, diagram, like dissection and understanding. You basically have to learn these animals in and out and how they work. And it's very interesting to have that experience and learn that, or know that this is going to be uh, useful in the future if we do decide to continue with studying similar types of phenomena with other animals. It was very eye-opening to see how much background work goes into these projects.
Okay. Well, I'd like to say kudos to all of you. You did a marvelous job. And it's kind of a throwback to when I first started doing research many decades ago. I won't say how many, but I still do a little bit, not myself, not personally, but my students. I still have a small lab at Cal State LA, not a group of 20 like I used to, but a small group of about 3 or 4 students. So once you have that bug in you, if you continue on in the STEM area, it's actually exciting if you never lose that interest in discovery. So congratulations to all of you. A virtual clap to everyone. Best of luck to all of you in your future studies. To those in attendance, I appreciate your supporting of our CSU's best and brightest. And I'd like to give kudos to Monica Alarcon for helping to assemble today's program. On that note, to all of you, Monica, do you have any final last words before I say adieu to everyone?
Sure. Just thank you to all the students. Your presentations were outstanding. Your dedication, creativity, and the depth of research that you showed in your presentations was truly inspiring. So I hope to see more of that in the future for all of you.
Take care, everyone. Goodbye.
Bye-bye.